All right, hi everybody. We are going to be talking about transport today in cells. And this might be a long one, I'm not sure, so you can speed up my talking by playing it back at a faster speed or plan on taking a break and coming back to this on another night. And if you're wondering, I am wearing a blankie because it is December and the room where I am doing the speaking is very cold. So I'm going to go ahead and get started. One unfortunate thing about this set of notes is that it's not in the same order as your textbook. So you're looking at like 5.2 through 5.5, and the notes don't follow it in order. So I just want you to listen to these notes, take notes, and then read it after that. So we're going to go ahead and get started. Transport is what we're talking about. Big questions that we're looking at. How does the cell control what is transported at the cell membrane? Why is transport of materials between the cell and its environment necessary for life? How does the environment influence living systems? And how do cells exist within the confines of the laws of thermodynamics? What is that? Okay, let's talk. So, all things being equal, things, very technical term, spread out. So we're going to talk about that specifically. So we mentioned the second law of thermodynamics. So the second law of thermodynamics says that the entropy of a system, which is sort of the randomness or the chaos or the disorder of a system, always increases. So that's puzzling because biological systems are extremely ordered. So how did biological systems come about? Well, look at the last part. It says, unless energy is added to that system. So, biological systems, plants are absorbing the sun's energy and they use that energy in order to maintain order, in order to organize things. So, like your bedroom has a tendency to get disordered, that's what this picture is showing. You have to put effort into it to get it to be ordered. You have to add energy. And so, it's the same thing that's happening with life forms. All right, so the first thing we're going to talk about is passive transport, then we'll talk about active transport. Passive transport, so transport, we're talking about the movement of molecules across the cell membrane. Passive transport, the phrase passive means without energy. It just means it's passive as opposed to active. So active using energy, passive is being relaxed, sort of. And passive transport always goes from where there's a high concentration to a low concentration. You may have learned about, like in junior high, like the concept of diffusion, which is a related concept, and we're going to talk about it. But that's not across a cell membrane, and that's how what we're going to talk about now is different. But remember that diffusion is like if I spray some perfume over in this corner of the room, because those molecules have because they have kinetic energy, because they have temperature, a measure of kinetic energy, they're going to diffuse, they're going to disperse throughout the room. They're highly concentrated here, but eventually the concentration will hit equilibrium as it spreads out throughout the room. And I didn't have to add any energy to it. It does it automatically. Same thing with this passive transport across cell membranes. So now let's focus in on how this happens in cells. So diffusion in a cell is the passive transport of molecules across a semi-permeable membrane. Semi means so-so, and permeable means that things can pass through it, like it has little holes in it or something. It doesn't. But semi-permeable means it's a little bit permeable to some certain kinds of molecules. All right, so diffusion is, he says here, inescapable as long as you can fit through the membrane. So diffusion automatically happens unless the membrane doesn't allow it because the molecule is too large. So take a look at this particular diagram. In the U-shaped tube at the bottom, there's a semi-permeable membrane. It's shown as like gray dashes, and there's a space in between each of the dashes that's a certain size. Notice that the sugar molecules, which are shown in green, are way too big to pass through those dashes, so they can't move from one side to the other. What's not shown in this diagram are the water molecules, and the water molecules are itsy bitsy and they can pass through those little pores. So what's going to happen is that the water molecules are going to move from where they're more concentrated to where they're less concentrated. So you're going to see the diffusion of water going from the left side to the right side. Now, let that make sense to you for just a second. On the left side, let's say that there are 10 sugar molecules in 20 milliliters of water. I don't know that. So 10 sugar molecules in 20 milliliters of water. But on the other side, let's say there's 20 sugar molecules in 10 milliliters, no, in 20 milliliters of water. So it's the same amount of water, I guess. So the water is there's fewer water molecules on the right side because the sugar molecules took up space. So there's not as much water on the right side as there is on the left side. Let me say that slowly. The sugar molecules on the right side are taking up space. So there's not as much water on the right side of the U tube as there is on the left side of the U tube. So what ends up happening is that water starts going from where it's more concentrated, which is on the left side. There's more water molecules on the left side of the tube, and they start gradually diffusing or osmosing across the semi-permeable membrane to the other side until it reaches the same concentration on both sides, which is called equilibrium. So now on the second U tube, so this is when it's all done, you see maybe I'll just make up numbers. Now it's roughly 10 sugar molecules for 10 milliliters of water. And the other side is 20 molecules of sugar for 20 milliliters of water. So the concentration is the same on both sides, and that was achieved by the water moving across rather than the sugar. The sugar wasn't able to move, so the water molecules could squeeze in between the pores of the semi-permeable membrane. So that's called osmosis when water is moving. The diffusion of water is called osmosis. Here are a couple of examples of diffusion. So in this first example, we have water on one side of the semi-permeable membrane, and we have molecules of dye that are small enough to diffuse. And so what we see is that there's random motion. Some of them make it through, some of them hit the barrier and return back, but the net movement in the first and second picture is across to the right side. By the time we get to the third picture, notice that the arrows going to the left and arrows going to the right, there's the same number of them. And so that's called equilibrium. Some of the color molecules, some of the molecules of the dye are going to move to the left and some are going to move to the right. The images previous to that, the primary wrong syllable. The primary motion was from left to right. And by the time we get to the third image, it's equal. It's moving both directions. In the second image, we're looking at two solutes instead of just one. And so they're both diffusing across until they both have achieved equilibrium. So notice that the yellow dots, I don't know what they are, the yellow dots on the left side are very concentrated on the left, but by the time we get to the third image, there are equal numbers of yellow dots on the left and the right, and same thing with the purple dots on the left and the right. Diffusion is what they call an emergent property, and I'm going to take just a second to explain what an emergent property is. It's a concept that's super important in AP biology, and it's very, very elusive for most students. They don't understand what emergent properties are. So I'll just start with a real simple explanation and just go with it. And then the more we get introduced to emergent properties, the more it'll start to sink in. An emergent property is a property that you only see in the larger perspective, and you don't see it in the component parts. So an example, I saw this online, I thought, oh, that's a perfect example, is a heart that can pump blood. So when we look at the whole heart, we can see that the heart is capable of pumping blood. But if we look at the component parts of a heart, the heart cells, no heart cell can pump blood. That's not what they do. But the entire thing together, the emergent property is the ability to pump blood. That's something that the tiny little parts cannot do by themselves. So diffusion is considered an emergent property, and I think this is confusing because if you just look at any one molecule, you don't really realize what's happening. Any one molecule, it's just moving randomly around. You have to step back and look at all of the molecules, and then you see that they're moving from a high concentration to a low concentration. So then that's an emergent property that you can see the whole process. Any one molecule isn't going to be isn't going to be diffusing all by itself because it's not going to be switching from high to low concentration. You need all of the molecules for there to be a high concentration and a low concentration. I don't know if that made sense, but it's a difficult concept. All right, so moving on. So we have to take into consideration chemistry with all of this. And this is why we have the biochemistry unit first in this class. So there's a couple questions we're going to take a look at. First of all, what can diffuse across the lipid bilayer, and then what do all the other things that are in the lipid bilayer do? So a couple things that we want to mention are the difference between channels, which we're going to talk about right now, and then the carrier proteins, which are coming up in just a second. So aquaporins, I love this name. I love easy names. Like the word aquaporin sounds a little bit intimidating, but if you break it down, aqua, I think most of you know, means water, and porin literally means it's a pore, it's a hole. And so aquaporins are these gaps, these holes in the lipid bilayer. So this pink area on the screen, that's the lipid bilayer. And it's literally a hole or a well that passes all the way through both layers that allows water molecules in and out of the cell. An ion channel is exactly the same thing, but for ions. So charged particles can travel through an ion channel, and nothing special really happens. They can just travel through. So those are a particular type of transport protein. So then, oops, my picture is in the way. So channels and carrier proteins control the diffusion of charged and polar molecules. So charged and polar molecules are an issue because they can't pass through the lipid bilayer on their own. Nonpolar molecules can. Nonpolar molecules can fit in between, fit is a funny way of saying, but can pass in between the nonpolar tails of the lipid bilayer. So nonpolar molecules can pass through, and really small polar molecules can pass through. Actually, water is one of the small polar molecules that can pass through. And let me just show you really quickly. If water can pass through, why do we have the aquaporins? And I was going to try to make this big. This is such a weird image. I might need to start it over. I was hoping I could make it big. It says it's unavailable. I see. Okay, well, let me show you again. So starting at the beginning, it's a cool video if you can see it here. This is a water molecule, and then this is they're the heads of the lipid bilayer. Here are the two sets of tails of the lipid bilayer, and then this is the other set of heads of the lipid bilayer. Watch that water molecule trying to pass through the lipid bilayer. It's a super cumbersome, crazy path that it has to go to, and I'm not even going to show you the whole thing. Give it a moment that you can watch how difficult it is for it to pass through to the other side. So this is what we need aquaporins for. Aquaporins are passages that the water can get through quickly. So yes, water is a small polar molecule that is capable of passing through the lipid bilayer, but it doesn't do it very efficiently. All right, back to where we were. All right, so there's a couple things that I want to be clear. The difference between a channel protein and a carrier protein, and we're going to talk a lot more about carrier proteins in just a second. A channel protein is basically like a hallway or a hole. It just allows molecules to pass right through. So there's not a whole lot of blocking it or stopping it. A carrier protein is a protein that actually changes shape. And we're going to see, I'll show you some videos on this in just a minute. But the whole maybe I will show it to you right now. No, I'm not going to show it. I'm going to show it to you later. But the entire protein changes shape. So right now that protein is open to the bottom, but once it releases the blue molecule, then the shape goes like this, and it opens to the next blue molecule. The blue molecule gets tucked in, and then it changes its shape. And this is an example of a carrier protein, where the carrier protein actually brings a molecule across, usually one molecule at a time, but sometimes it can bring a few molecules across. So how can we control diffusion? That's where active transport comes in, and that's the next part. So active transport, as the name implies, is the transport of molecules that requires energy. We're going to be going against diffusion. So instead of going from high concentration to low, like going on a sled hill, going from the top of the hill to the bottom, doesn't require a whole lot of energy. Now we're going to go up the hill with our sled, and that requires energy. We're going to go from low concentration to high concentration. That's called active transport. and it uses a protein because the protein is what's going to be accessing the energy in the ATP molecule that's going to power this transport process. So I need you to get really good at studying these diagrams. These diagrams definitely will show up on tests and probably without some of the labels. So you need to understand what's happening here. So you see the lipid bilayer. The purple little oval in the middle is a protein. In this case, it's a proton pump. That's its job, is to move hydrogen ions. Remember, you can call them hydrogen ions or you can call them protons. You can move them, it moves them across the lipid bilayer. But what I want you to really pay attention to is the fact that normally things diffuse from high concentration to low. So they should be going from the extracellular fluid, the liquid outside of a cell, extracellular fluid outside of a cell. They should be going from the outside of the cell to the inside of the cell. But this pump is forcing them to go from low concentration on the inside of the cell to high concentration on the outside of the cell. Well, the only way it can go the opposite of diffusion is to use up some of the cell's energy. The cell's energy is called adenosine triphosphate. We're going to learn a little bit more about that later. But that energy has to be used in order to pump that proton across. So what we're looking at is a form of active transport. Now, this next one shows why we're actively transporting it across. So there's that proton pump again. But now notice the cell purposely wanted a high hydrogen ion concentration on the outside so that those hydrogen ions could act as a co-transporter of sucrose. So we're trying to get sucrose into the cell. Notice it's a disaccharide, trying to remind you of some of your vocabulary. We want the sucrose to come in. The other protein that allows sucrose to come in requires hydrogen ions to assist the sucrose to come in. So if we have lots of hydrogen ions on the outside, then we can bring lots of sucrose into the cell. So that's an example of co-transporters. All right, so where do we get the energy for active transport? We get it from ATP, and I'm going to show you a little video in just a second. So trying to see phosphorylation. Let's just talk about that word really quickly. It sounds like a scary word. ATP stands for adenosine triphosphate. You've seen this molecule before. You saw it in our biochemistry unit. It looks like the building blocks of DNA, only it has 3 phosphates instead of just 1 phosphate molecule. So phosphorylation refers to, let's see if we can see it on this diagram here. Phosphorylation, there's ATP and one of its phosphates. When it leaves behind, when it removes one of its phosphates, energy in essence is released in the form of a chemical bond. Energy is released. It causes the purple protein. Look at the protein, how it changed shape here once the ATP lost one of its phosphates. The shape of the protein changed, and that's what's going to allow sodium and potassium to pass through. And I'm going to show you a video on that here really quickly. So that's called phosphorylation, and it's what triggers the conformational change, the shape change in the protein. And yeah, so let's go ahead and watch a video. I don't remember how long this is, a couple minutes, 2 minutes and 26 seconds. I might speed it up if I think it's too slow.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate.
Phosphorylation.
This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since
One of my favorite things about biology and how these things all work is the way that it recycles itself. So we think that we invented recycling when in fact we invented waste and we're just rediscovering recycling. This planet recycles and your body recycles. So look at that ATP molecule, how it dropped off a phosphate, released its energy, and then when it's done, the phosphate gets released. Guess where that phosphate goes? To get reattached to an ADP, adenosine diphosphate, to go back into adenosine triphosphate. So we're just going to keep recycling that ADP and ATP molecule going around and around. And I love that. I think that's super cool. All right, so hopefully that little video was clear about what they call conformational change, when the shape of the protein, the carrier protein, is changing shape. That one required adenosine triphosphate to happen. All right, moving on. Tonicity. That's such an ugly word. We're talking about hypertonic, hypotonic, and isotonic solutions. Tonicity is a relative measure of how concentrated a solution is. The tonicity of a cell's environment has serious consequences for the cell. And I'm going to go into a lot more detail right here than you would expect. So take a look at these diagrams. Super, so many important concepts in biology right in this one little diagram here. So first of all, a hypotonic, you need to have, I should say this, you need to have a piece of paper and pencil out. If you didn't already, you better have had them out already. But you need to draw this diagram. You need to write the words hypotonic, isotonic, hypertonic. You need to write animal cell and plant cell. You need to circle the ones that are good. And you need to write examples of hypo, iso, and hypertonic solutions. So now I'm going to start again, and I want you to be writing like a crazy person. So a hypotonic solution is any solution that has a lower concentration of dissolved substances than what's inside of the cell. That's confusing because most of you don't know the prefix hypo. But if we were to jump over to the other side and say what a hypertonic solution is, that one makes maybe more sense intuitively because we think of words like hyperactive and we understand that that's overactive. So a hypertonic solution is one that has more dissolved substances than what's inside the cell, a higher level of dissolved substances than what's inside the cell. Dissolved substances are called solutes. What they're dissolved in is solvents. That's just a reminder. You should know that from chemistry, but just reminding you of it. Okay, so now let's talk how that impacts the cell. A hypotonic solution, a really easy example of a hypotonic solution is distilled water. Write it down. Okay, what percent water is distilled water? Well, duh, it's 100% water because that's all distilled water is. It doesn't have anything dissolved in it. So it's 100% water. So that means it has 0% dissolved substances. And what that also means is that the water is absolutely the most concentrated it can possibly be because there's nothing else in it. There's tons of stuff dissolved inside of a cell. So the concentration of a cell's cytoplasm is about 80% water. So we said that diffusion, or osmosis in this case, is where water moves from a high concentration to a low concentration. So outside of the first cell where it says animal cell in a hypotonic solution, we're imagining that that's distilled water, 100% water. And if you want to, in your drawing, you could say that inside of the cell, it's 80% water. So which way is the water going to move? It always moves from the big high number to the low number, from high concentration to low concentration. Ooh, I should say because this is passive transport. Remember, diffusion and osmosis don't have energy. They don't require energy. They don't go across the concentration gradient. They don't go the wrong direction. They always go the right direction, like a sled going down a hill. So they're going to go from high to low concentration. Okay, here's my point. Water, if you take this blood cell and you put it in a dish of distilled water, there's so much water on the outside compared to the amount of water that's on the inside that the water is going to start rushing into the cell. And if it's an animal cell, and eventually that cell will pop. So distilled water, super bad for an animal cell. And we'll get to the plant cell in just a second. Ideally, what an animal cell, a blood cell will say, wants is an isotonic solution. The prefix iso means same. Hypo means less than, iso means same, hyper means more than. The reason those prefixes can get confusing is that they're talking about the concentration of dissolved substances, not the concentration of water. So let's practice that for a second. I told you that distilled water is 100% water. That's as high a concentration of water as you can get. But the prefix is hypo. That's because hypo is referring to the concentration of dissolved substances, which is super low, 0. There's no dissolved substances in it. So that's why that's a hypotonic solution. An isotonic solution has the same concentration of dissolved substances outside as inside. So in our example, we said that the cytoplasm of the cell was 80% water. So let's imagine a scenario, write this down, 80% water inside the cell, put an 80% water outside the cell. What you're going to see is some water enters, some water leave. That's called equilibrium, when it comes and goes at an equal rate. And that's the ideal for an animal cell. The cell stays the same size. That's what the animal cell wants. And the animal is going to do whatever it has to make sure that that stays correct. So for example, an animal is going to drink and it's going to pee to make sure that the concentration of water inside its body is correct. And when we're not drinking well, then our cells aren't able to function properly. So that's why that's one of many, many, many reasons to drink well, to drink water a lot. Okay, so now our third example is a hypertonic solution. What I recommend you write down for your example for a hypertonic solution is ocean water or salt water. So now let's imagine a concentration. It's a high concentration of dissolved substances. So let's just pretend that it's like 30% salt, which means what percent water? If it's 30% salt, that's that high hyper concentration, then it means it's only about 70% water. So there's very little water in comparison to distilled water. There's very little water in salt water. Let me give you another example. If I have 2 cups, I have a cup of distilled water and I have a cup of salt water. Which one has more water in it? Even if it's the same amount, it looks like the same amount. The one that's distilled water, I can't remember which hand I did. The one that's distilled water has way more water in it because it's only got water molecules. It doesn't have anything dissolved in it. The saltwater one has way less water in it because there's all these salt ions, all the sodium and chlorine ions in it. All right, so in a hypertonic solution, the percentage of water on the outside, hopefully you wrote down 75% water, it's lower than it is inside the cell, which was still 80%. So water is always going to travel from high concentration to low concentration. And so the high concentration was in the cell, 80%, and it leaves the cell to try to balance out the concentration so that it evens out. So now let's take a look at plant cells. Plant cells are different for a very key reason. They have the cell wall. They have a cell wall. I know what it's called. They have a cell wall. You have a skeleton. So your cells are all soft because you have a skeleton that holds you up. Plants have a cell wall because they don't have a skeleton. So now if you take a plant and you put a plant in distilled water, so that's our hypotonic solution, water is going to start rushing into that plant cell. However, the plant cell is not going to pop like an animal cell would because plant cells have this rigid cell wall on the outside. So what ends up happening is that the pressure inside the plant cell starts to increase, only to a certain point. and it increases, and that's actually good for the cell, for the plant. That allows the plant to stand up and photosynthesize, and it's all good and healthy. You need to write down turgid, so that means good pressure, or you could say high turgor pressure. Turgor is Turgor. So high turgor pressure inside the plant cell, and you get that from a high water concentration on the outside. So distilled water is actually good for plants if it's got a nice high water concentration that's coming into the plant. All right, so in the next example, that's an isotonic solution, and that means that the water is coming and going at an equal rate, and the plant cell is okay, but it doesn't have this good water pressure that really allows it to stand upright. So this is where the plant is starting to get a little bit wobbly. Maybe it's not doing very well. Our cells would be happy as could be, plant cells not necessarily. So now if we switch and we take that plant cell and we put it into a hypertonic solution, say some kind of salt water, we put that plant into salt water, the water is going to be more concentrated in the cell, in the plant cell, than it is outside. So it's going to leave the plant cell. That's going to cause the cell to lose its turgor pressure. The turgor pressure will go down. We call it plasmolyzed. I have to think about what that word means, and I don't have it off the top of my head. I mean, I know what plasmolyzed means. I was going to try to break down the word, why it's called plasmolyzed. But anyway, it means that it's going to become floppy now. And so if you were seeing like a flower that is plasmolyzed, where its cells are plasmolyzed, it's wilting. So it's bent over, and it desperately needs to be watered. And once you water it, then the water goes back in, increases that turgor pressure inside, and then the plant can stand upright again. So there are some super important concepts here that are super interesting, I think. One of them is, why do you not drink, if you get stranded on a boat, we've all heard this story, if you get stranded on a boat in the middle of the ocean, you're not supposed to drink the ocean water. Why not? Well, put that in context here. Why are you not supposed to drink the ocean water? Give you a moment to think about it. Which of these situations is you drinking ocean water? And the answer is the hypertonic solution. So when you drink ocean water, this is weird, but the concentration of water in the ocean is less than the concentration of water in your cells. What? That's crazy, KJ. Right, it is. But that's the way it is. There's so much salt in ocean water that if you drink ocean water, it actually causes water to leave your cells and leaves you more dehydrated than if you hadn't had the water in the first place. That is too crazy for words. All right, so that's why you don't drink ocean water. Now, the other thing that I think is more important, because, how often does that happen, that we get stranded on a boat in the middle of the ocean? Hopefully never. But what is happening, hugely important concept in Minnesota, I need to switch screens here so you can get this. Come on, screen. I'm trying to get something to work and it's not working. Oh my gosh, are you serious? It didn't work. Oh, give me a second. That's why I needed to open my screen up a little bit. So I'm going to click on this really quickly, and I want to just show you what's happening in Minnesota, because it breaks my heart and it makes me cringe. And we need people to speak out against what's been going on in Minnesota. Every single winter, our roads get icy. So what do we do to our roads? We add salt to our roads. Now, that's not meant to be a cell or anything. That was just a circle around it. So we add salt to our roads. Salt doesn't evaporate. Salt never goes away. Never goes away. Salt never goes away. So we add salt all over our roads. So what happens to that salt? Well, in the spring, the snow melts and the salt gets carried into our sewer systems, and then into our, it's like our street drainage systems, and then washes into our lakes. It also gets trapped in our grass, blah, 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 blah. So who cares, KJ? What do I care? So let's talk about what's going to happen to plants. I've drawn a plant cell right here. Let's say that the plant cell, I made this number up. It doesn't matter what the number is, but let's say a plant cell normally has 5% salt inside of it, which means that the plant cell is normally 95% water. If we add salt outside of plants, so if I add salt to my sidewalk and then I try growing grass the next year, the water on the outside of my grass is going to be 75% water and 25% salt because I added all this salt in the winter to melt the ice. So what does that do to my grass? Well, it causes the water to leave. Water went from a higher concentration, 95, to a lower concentration, 75. And my plants go, they're plasmalized, my grass dies, and that's bad for my grass. Who cares about my grass, KJ? You guys probably don't care about my grass. However, what you might care about is the fact that this is now all washing into our rivers and our lakes. So now what's happening to our lakes? Well, we have fishies that live in those lakes. And now those fishies have, I'm going to make up a number, we'll say 80% water in those fishies. And as the salt levels increase in our lakes, that causes water to leave our fishies. They desiccate and they can die. Plant life dies, all kinds of things. So it's super, super harmful. The fact that we have 10,000 lakes in Minnesota, the fact that we're adding so much salt into our environment, and there is no way to remove that salt. So every year that we're adding more salt, we are making this environmental disaster worse and worse and worse. Okay, I really actually feel anxiety about that particular topic. So we need to figure out other things. Oops, what did I screw up? We need to figure out other ways to melt ice other than salt. And it needs to be chemicals that are safe for the environment. Okay, where was I, you guys? I don't even know where I am. Moving on. Oh my gosh, how can I get there as fast as possible? I wasn't that far, was I? Oh, I was that far. Okay, let's keep going. I'm sorry. You can fast forward through this pokey part. Okay, so we are right here. I'm going to actually do another diagram here. So let's just do a problem. You're going to need to solve problems on tonicity for any test for sure. So I'm going to give you an example. Here's a beaker, and there's a pretend cell in this beaker. This is like a semi-permeable membrane, but we'll just say it's a cell right now. And in our cell, we're going to say it has a 0.03 molar sucrose solution and a 0.02 molar glucose solution. Now, right now, you guys don't know what molarity is. You haven't learned that yet in chemistry class, so no big deal. Let's instead, this totally works as long as we're consistent. Instead of saying it's a 0.03 molar solution, let's say it's a 3% sucrose solution and a 2% glucose solution. So about 5% of our liquid is made of sucrose and glucose. So what percent is then water? Figure that out yourselves. Now in the environment, in the blue part of the beaker, we have 1 molar sucrose. So we'll say 1% sucrose, 1% glucose, and 1% fructose. And then the rest of it is water. So let me pull this up quick and go to the next page. Okay, so this is supposed to be the same diagram. I hope you can follow my diagram. So if we have a 5% sugar solution, we have a 5% sugar solution, that means the cell is 95% water. Oops, that's H2O. All right, and in the environment, that's out here, we have, this is a 3% solution, 3% of dissolved substances. So that means we have 97% water. So what's going to happen to our cell if it's got 95% water out inside and 97% on the outside? Water always goes from high concentration to low concentration. So the water is going to go from the outside into the cell. The cell is going to gain mass, and if it's not tough enough, then it's going to burst open. The cell's going to break open. Bad situation. So then here's another question: is this a hyper, hypo, or isotonic solution? Well, it's for sure not an isotonic solution because we know iso means that the water is going to come and go at an equal rate. So it's either a hyper or a hypo solution. So hypertonic is like salt water. It causes things to shrivel up. And hypo is like distilled water, which causes things to swell. Now, I had a student a number of years ago tell me, and I thought this was just brilliant. This is how she remembered what happens to in a hypotonic solution. She said, "Hey, KJ, when I hear hypo, I think of the African animal, the hippo. And a hippo is big and swollen and humongous. So that works, and I've been teaching it ever since. So if it's getting bigger, the volume is increasing, then it's in a hypotonic solution. Water is rushing in, and that's making it get bigger. All right, back to where we were. Make it full size again. Okay. Next, there's another video I want to show you. So contractile vacuoles, this is a little single-celled microscopic organism that's called Paramecium. Paramecium has a built-in vacuole that's called a contractile vacuole because it can contract and push water out. And I just wanted you to see it quick for like 5 seconds, and then we will move on. The contractile vacuole, you'll see it in just a second because you'll see it close up. So look at it. And it closed up. So watch it one more time. It'll fill up. There it is, getting bigger and bigger again. And then watch, and it'll go whoosh. Give it a second. And whoosh, whoosh. Come on, do it there. And it pushes water out. So that's a way for this little animal. It's not really an animal. It's a protist. That's a way for this little animal. I'm going to say animal anyway. To survive in a hypotonic solution. So if water is rushing into it, it can collect the water and push the water out. So it can survive in a hypotonic solution. However, the pump only works in one direction. So if you leave it in a hypotonic solution, it takes ATP. So the paramecium is going to get tired. It's going to have to keep eating and work hard. So it really wants to be in an isotonic solution, not a hypotonic solution if possible. But if we move this paramecium, which I would never do because they're so lovely, if I were to move a paramecium into a hypertonic solution like salt water, its pump only goes one direction. It can only push water out. Sitting in salt water, the water would leave it and the little guy would desiccate. So its pump would stop pumping and water would just start oozing out of its body and it would die. I never let that happen. All right, so next we need to talk about fishies, because how is it that fish can live in saltwater? What is it that they're doing differently than, say, freshwater fish? If we take the fish from I have a huge 55-gallon tank in my classroom. If I take my freshwater fish and put them in the ocean, they will die. Why is that? So saltwater fish have some adaptations that allow them to survive in saltwater. So take a look at the arrows. The dark blue arrow represents salt, and it's a marine fish, a saltwater fish on the left, and a freshwater fish like we would have in Minnesota on the right. The light colored arrows represent water, and the size of the arrow is relevant. It's showing you how much is coming in or coming out. So what happens to a fish in saltwater, just like what happened to you in saltwater, is that water starts leaving the fish because it's got a higher concentration of water in its body than the ocean does. So water has a tendency to leave it. So the fish does several things to adapt for that. Number one, when it's eating, it gets water in when it's eating. So that brings water in. But it also drinks a lot of water, a lot of the ocean water, to replace the water that it's constantly losing. So it's this constant in and out water. It's excreting some of the salt from its gills to help with this saltwater balance, and it loses a lot of water out of its gills. But another way to adapt, so it's bringing in a lot of water in its mouth, it's losing a lot of water out of its gills, but another adaptation is that it barely pees. So hardly any water gets lost through urine. That is in contrast to a freshwater fish. Freshwater fish don't drink in lots of water. They get some water when they're eating. But the water that they're in has a tendency to move into their bodies. So they tend to, it's a hypotonic solution for those fish, and water has a tendency to make them bloat. So they get rid of tons of water by urinating a lot. So they pee into the water. That's a pleasant thought when you think about swimming in our lakes. But anyway, they're getting rid of a lot more water in their urine than the saltwater fish are. Okay, moving on. Passive transport versus active transport. This is just an image to sort of make sure you understand what's going on. I would recommend drawing this image. You don't have to make it anything beautiful, but just have this as a drawing. So I could totally see myself using this drawing without the labels and expecting you to be able to put the labels on. So on the left side are all passive examples of passive transport. I can tell, even though it's labeled, like if it weren't labeled, because there's no ATP in the left-hand images. So I know that those are all examples of passive transport. In the first example, we see diffusion. We see molecules that are diffusing across the lipid bilayer without any help. That's an important word, without any help from proteins. The next two diagrams at the bottom, it says facilitated diffusion. To facilitate something means to help it along. So these are when diffusion is happening and it's being helped along by either on the middle diagram is a channel protein that's just a hallway that allows molecules to move across, or a carrier protein on the other on next to it, which is one of the ones that changes its shape and allows molecules to move across. But that one, notice carefully that that particular carrier protein does not require ATP. It's changing conformation on its own. Now, the last one is the active transport, where molecules are moving across the concentration gradient, opposite the concentration gradient, and it requires ATP. So ATP is being used, so I know it's active transport. All right, let's move on. Last thing, we're almost done. This last section is called bulk transport, and we've actually already seen this. You guys just haven't had names for this yet. So bulk transport, as its name implies, is moving large molecules. If the molecules are moving into the cell, it's called endocytosis. If the molecules are moving out of the cell, it's called like exiting, exocytosis. Endo in, exo out. So in endocytosis, there are three types of endocytosis. The first type is referred to as cell eating. It's called phagocytosis. And this particular diagram cracks me up because it looks like it's eating a turd to me. That's what the diagram has always looked like to me. So apparently that's not poo, that's food, but it's something that the cell needs. So it's going to form a food vacuole around it, not unlike what they think happened when prokaryotes were absorbed by eukaryotes with the endosymbiotic theory. So that could have been like a mitochondria that just got absorbed, for example. Anyway, so that's food on the inside. That food vacuole will then fuse with a lysosome, and the lysosome will digest the food on the inside. I'm trying to think if there's anything else you need to know about that. So that's called phagocytosis. Cytosis refers to eating, but I don't remember what phago means. Is it food? Not positive on that one. Pinocytosis is the next one. You can see the word off on the left side. And it's called cell drinking, but it's basically little vesicles forming on the inside, and it's a way for it to bring in small molecules, especially molecules including molecules of water, but just smaller molecules are being brought in. And then the last one, which is called receptor-mediated endocytosis. Ooh, it sounds fancy. And this is where the author says it's the cell being picky. If you look at those little Y-shaped receptors on the cell, those Y-shaped receptors are like little magnets for a very specific type of molecule. In this case, it's a purple triangle. And they're like magnets. They're attracted to each other. It's polar, et cetera, et cetera. They're attracted to each other. So they pull in the molecule that the cell needs. So molecules in the environment, you have these little receptors, and they come in, and then then they get pinched into a vesicle, and then that vesicle gets carried away to the cell, maybe to a lysosome. It depends what it is and how it's being used. But that's called receptor-mediated endocytosis. All right, and then the very last thing, and then we're done. This was a long one, I warned you. It's called exocytosis. It's when vesicles merge with the cell membrane and they release the product. It could be waste products, but it could be very important things like a hormone, perhaps, is being packaged in the Golgi and then being released out of the Golgi. So it's the release of large cellular products to the cell membrane. That is it. Any questions? Let's go over a couple quick things. I'm going to let you just look at this. These are good things to be able that you should be able to know. And if you have any questions, you can present them to me in class. You can pause it here if you want, and you don't need to do that. And we are all done, cuties. Later. Oops.